Now, after learning about the types and characteristics of dendritic cells, in this section, we will look at the functions of dendritic cells. And the main function or role of dendritic cells is in antigen presentation and T-cell activation. So, what happens is that when tissue is infected by a microbe, the microbe will initially encounter cells of the innate immune system, such as tissue macrophages and other leukocytes. This may initiate an inflammatory response. Another cell type found in tissues play a vital role here in stimulating adaptive immune response by stimulating CD4 T cells. And these cells are none other than the dendritic cells. And we know that DCs are bone marrow-derived cells that are found in almost all tissues, mainly in lymphoid tissues, but also in non-lymphoid tissues. Dendritic cells in non-lymphoid tissues are also present and are called tissue dendritic cells. Like Langerhans cells are a specialized type of tissue DCs found in the skin. Dendritic cells are formed from hematopoietic progenitor cells in the bone marrow. Initially, these progenitor cells form immature dendritic cells that have the endocytic capability, but a low capacity to stimulate T cells. These immature cells keep on monitoring their environment for invaders, such as bacteria and viruses. And they achieve this through pattern recognition receptors, PRs, such as the toll-like receptors, TLRs, that are present on their surface. As soon as these immature cells come into contact with the antigen, they mature with extended projections and thus move toward the lymphoid organs. The immature dendritic cell engulfs the pathogen and breaks down its proteins, which it presents on its surface using MHC molecules once it matures. At the same time, the cell surface receptors that act as co-receptors in T-cell activation are upregulated by dendritic cells. Dendritic cells are highly efficient at taking up antigens by pinocytosis, which is the process of uptake of extracellular fluids and dissolved solids, mainly antigens, or through receptor-mediated endocytosis, which is a form of endocytosis or inward movement in which receptor proteins on the cell surface are used to capture a specific target molecule. And the receptors involved here are usually mannose receptors. Or another process involved here is phagocytosis. That is the internalization or engulfment of particles larger than 0.5 micrometers in size within a vesicle called a phagosome. So, by any of these mechanisms, antigen uptake is done by dendritic cells. Now, once antigen uptake is done, and they are activated, DCs are stimulated to leave the tissue site and enter the draining lymphatic vessels, in which they will be carried to the local lymph node. This migration of dendritic cells to the lymphatic vessels is induced by a chemokine called the Epstein-Barr virus-induced receptor ligand chemokine, or ELC. This ligand binds the chemokine receptor, CCR7, which is upregulated on the dendritic cells at the time of their activation. As they migrate from the tissue to the lymph node, the DCs process the antigen and express antigen fragments on their class II MHC for recognition by CD4 T cells. Once in the lymph node, the dendritic cells migrate to the T cell area, the paracortex, where they can encounter CD4 T cells, as they are already in abundance in this area. Also, antigens derived from microbes at the site of infection can also enter the lymphatic ducts in free form and be carried to the local lymph nodes. As the antigen percolates through the lymph node, it is picked up by dendritic cells in the paracortex, where it can be processed and antigenic fragments presented to T cells on class II MHC. In this way, antigens are presented on the surface of dendritic cells and are thus presented to the helper T cells. These helper T cells then further play their role in adaptive immune response by activating B cells for the production of antibodies against a particular antigen. They help in the activation of macrophages for the engulfment of particles. And they also help activate further cytotoxic T cells that kill infected target cells. So, this was all about today's video. For more such educational videos, keep watching sqadia.com.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.